organizations. This is the Venture Institute campus in Rockville, Maryland. We also have a campus in La Jolla, and a few years back, I guess it's been 5 years now, Ham Smith and I started a new biotech company called Synthetic Genomics around the work we were trying to do with making the first synthetic species. And we just broke ground on the UC San Diego campus. We're trying to build the first zero-carbon research building in the world. So this building will generate all its own energy. We'll have zero net carbon, including the transportation to get there. And we're going to recycle all our own water in this building if you ever want to stop by for a drink of water. Find out whether it's good or not. So it's pretty exciting trying to put all the concepts together and at every aspect of what we do at the Institute. So obviously I'm going to talk a little bit about DNA tonight, and I want to convince you all that DNA is the key molecule of life. In fact, I view it as software. Hopefully by the end of this hour, you'll be convinced why it's software. When we read the sequence of a genome, the first bacteria you heard about was 1.8 million letters. Our genomes are roughly 6 billion letters. We have to get those in the right order, but when we go from this 4-letter code of ACs, Gs, and Ts into the computer, I call that digitizing biology. We're going from the AC, Gs, and Ts to the 1s and 0s. And that's been the challenge of the first few decades of genomics, was trying to digitize all this information. Now a fun challenge is trying to go the other way. Can we start with this digital code in the computer and get life out of that computer and out of that code? So let me walk you through that. We try to ask some big questions, having a little bit of freedom to do that. Ones that other people have asked. What is life? How extensive is it? Biology has been reductionist in practice, trying to pare things down to their minimal components so we can understand how they work and then put it back together. Obviously, can we digitize it? I just told you about that. And then trying to create new life out of that digital world. So back in 1990, as you heard, I had this idea for just isolating the messenger RNA in the brain. So when our genetic code gets read, it gets converted into a different kind of chemical code called messenger RNA. And that tells you which genes are expressed. And we found just by sequencing these, we could find which genes were in what tissues. It started off slowly, but you can see the numbers of these in the public databases have grown quite substantially over the years. And they're still growing fast. The problem was having all these sequences, and how do you interpret them, sort them out? So we had to develop all kinds of new computer algorithms and approaches. And one of these was a way to assemble these sequences together to try and see how many were unique. We thought we could use this new algorithm to maybe assemble an entire genome. So that's what we set out to do in 1994 and published this paper in 1995 and ended up being the first genome that anybody decoded. This was an exciting point in biology because we saw for the first time all the genes that represent life in one organism. And it's gone on to be a method now that's used pretty universally, the shotgun sequencing method, for understanding genomes, including the human genome. Now, the good news is technology is changing, and changing quite rapidly. At Celera, we had to have 300 of these $350,000 machines. This was the lab at the Venture Institute about 5 years ago. Just a huge data factory of turning the DNA code into digital code. But over this past decade, the technology has changed, and we've replaced this entire 60,000 square foot facility with a small benchtop machine. This is what we use for sequencing the human genome. There's all these machines, but now it's very simple. This is the team that actually led the program. Tall man in the middle is Hans Smith, who got the Nobel Prize in 1978. Gene Meyer, standing next to me, developed the algorithms for assembling the human genome. And Mark Adams developed all the all the automation. This was us just a short while ago in San Diego celebrating the 10th anniversary. All this proves is you look better in a tuxedo than in a suit. There's a fun event. So here's these new machines. They cost about $500,000 each, about the size of this podium, and replace these giant facilities. Everybody knows the same things have happened in computing. So in 1999, we built the third largest computer in the world. It seemed big at the time with 1.5 teraops. But now, instead of this giant room, you can buy a teraop add-on to your PC. So fortunately, things get smaller and faster. And that's going to be very critical if we're going to sequence everybody's genome. In fact, the challenge has been to get the technology fast enough, cheap enough, and accurate enough. So several years ago, we started the X Prize to create a contest for people to develop better and faster technology. My friend Stuart Lucen is here tonight. He actually put up the $10 million for the X Prize. For some reason, that motivated a lot of people to try and develop new technology. And it's been pretty exciting what that's leading to. And hopefully in the next couple of years, we'll be awarding that prize. So the human genome sequence, the first draft, came just 5 years after we developed these methodologies. But it took another 7 years for the progress in genomics to get down to individual genomes. So in 2007, we published My Complete Genome, and it changed our view of human genetics. In the year 2000, we only published half of the genome, in part because people thought it was going to be about $1 a base pair to sequence the human genome. And so what was settled on was just doing half of the genome, just one set of the chromosomes that was obtained from just one parent, because at 3 billion base pairs, that would be $3 billion, and everybody thought it would be way too expensive to have a $6 billion program. Like, when you're in the billions of dollars, I'm not sure it mattered a whole lot, but I think it looks far better now at $8,000 a genome, and soon to be, with the X Prize down below $1,000. But with this, we found that, in fact, my 2 sets of parental chromosomes differed by 0.5% from each other. In 2000, we thought we differed only by 1 letter out of 1,000 base pairs. So comparing my genome to any of yours, we find a 1 to 3% difference. And we're able to now, with some exciting new techniques, even sort the chromosomes and the sequence into the chromosomes we got from our fathers and our mothers. And one of the ways we do this is some exciting new technology. We can actually sort single cells. In this case, it was sorting sperm cells. We can isolate one single sperm and do the entire human genome on a single sperm cell. As I'm sure you all know from your biology classes or practice, sperm and eggs each only have a haploid genome. They have half the genome. When you put them together, you get in trouble. But by sequencing the genome and the eggs or sperm, we can get the half of the genome and work out what came from one parent. This is going to be important. Everybody's asked themselves questions about their family of, did you get your mother's nose and your father's ears? And now we can actually sort out some of that information as we go forward. Vanessa Hayes at my institute has been looking at human diversity, primarily in Africa, and some indigenous populations populations there, and coming up with some pretty interesting data. And also the Beijing Institute in China has used these techniques now to sequence the first Chinese genome. And we found, in fact, we do have this 1 to 3% variation across pretty diverse populations. But what Vanessa has found, one of the first genomes she did was Desmond Tutu's genome, who joined us for the 10th anniversary of the sequencing of the genome. There's more diversity between these different groups in Africa than between every African and the rest of the world, in part because of the long lineages there. She's now trying to trace this down to which groups really look like they migrated out of Africa, leading to the diversity of genomes on this planet. So it's exciting where we can go back and actually capture human history. Now, we're not alone. We have 20-some thousand genes in 100 trillion human cells. But you have approximately 200 trillion microbial cells associated with each of our bodies. That may not sound like a pleasant concept, but you have about 1,000 unique species in your mouth, contributing on the order of 4 million unique genes. So our 20,000 genes get swamped out by these roughly 10 million microbial genes. So just think about that when the next person sitting next to you is coughing or sneezing. They'd be sharing some of their genetics with you. But the challenge is, we always knew we had some microbes associated with us, but these numbers were a huge surprise. And this was done simply by our team applying the same shotgun sequencing that I'll tell you about for the ocean, what we did for the human genome, first to the human intestinal tract. So there's now a huge new field based on these early data that we had, trying to see what role these microbes play in human health and disease. I'll just show you one example. This is the change in various cancers since the mid-70s. And you can see esophageal cancer just really stands out, changing much faster than the others. And when we do the microbiome in the esophagus of these individuals, we find there's a set of microbes that actually totally associate with the esophageal cancers. We don't know yet, is this a consequence of the cancer or is it actually a cause? But esophageal cancer is one that's known that there are definitely environmental causes. The other question, what's the metabolic potential of all these organisms? What are they contributing? So one way to measure this is to look at all the chemicals in our bloodstream. Our 20,000 some odd genes make on the order of 300,000 different proteins, but we can make 2,400 to 2,500 unique chemicals. That's pretty good at you doing that without thinking about it. But it's a pretty finite set. So if we're going to take a blood sample from each of you right now, what would we find in your bloodstream? We find about 500 chemicals, but only about 60% of these are from our own human metabolism. So 30% are from all the things we eat in our diet. We absorb all these different chemicals from our food. But 10%, or about 50 chemicals circulating in your bloodstream right now through your brain, are bacterial metabolites of our own chemicals and the chemicals we eat. So we don't know what these do. Most people who study pharmacology thought there would be a few hormones and a few chemicals circulating in the bloodstream. Nobody had any idea. It was on the order of 500. On the West Coast, people always assume there's more circulating in the bloodstream. In general, that's true, but this adds substantially to it. We don't know what role these play. Do they cause disease? Do they make us feel better? Do they make us feel worse? So this is going to be a big challenge as we go forward. And this is called the metabolome. So we have all the microbiome, we have all the chemicals. So our complexity is getting higher and higher. actually gives me optimism because for the first time in our history, we know most of the fundamental components now associated with our lives here. So we have a chance to decipher it. Where in the past, ignorance of all these different components, there was no way we could ever get the right answer. Now, we use these same techniques roughly at the same time to ask questions about diversity in the oceans. And the notion was just to do a shotgun sequencing of the oceans. Environmental microbiologists thought there was a pretty limited subset of microbes, particularly in the ocean. We were told we'd find 20 or so unique ones in the Sargasso Sea off of Bermuda. So we decided to do the experiment, and this is my research vessel, Sorcerer 2. This really pisses off the guys in the Department of Energy that, you know, they have to work in acid mines to get samples. And we try to sail around the equator in nice places. Just shows we're smarter. So we used this and took some samples. And instead of finding 20 or so organisms, we stopped sequencing after discovering 1.3 million new genes that had never been seen before, just by taking what we find in the sea, filtering out, getting the DNA, and sequencing it. So we knew diversity was far greater than anybody imagined. And so we decided, instead of just sampling over and over again in Bermuda, to sample on a broader basis, and that's what led to the Sorcerer 2 expedition. The first major results were published in the special issue of PLOS Biology a few years back, with multiple articles looking at the diversity and the extent of that diversity. This is the route that we followed, very similar to other sailing expeditions. In fact, the Challenger expedition in the 1870s stopped and took samples every 200 miles. They were basically trying to see how deep in the oceans life went. So they sent a dredge down every 200 miles, take samples, and they were pretty excited because they discovered in a 5-year circumnavigation about 5,000 new species that people hadn't seen before. So I figured that was a good paradigm, so we took samples on this route. We covered both coasts. We actually sailed by here a couple of years ago on the way up to Glacier Bay in Alaska. So we sampled basically from Panama to Glacier Bay on the west coast and up to Halifax on the east coast. Then this last 2 years, we've been sampling first in the Baltic Sea. Then last year it was really tough duty in the Mediterranean. You know, I had to spend 6 weeks sailing around Italy and Greece and Turkey, and then we went up into the Black Sea. And so you can see there's a lot of diversity, but these sort of inland seas have been surrounded by humanity for a very long time. And what we're already starting to see is totally different sets of organisms there. Our protocol is very simple. We just filter seawater through these different size filters, collecting everything from large single-cell eukaryotes to single bacteria, and then everything that goes through this 0.1 micron filter we collect as a viral fraction that we assay independently. This is the apparatus on the back of Sorcerer 2. Jeff Hoffman, who did most of the sampling around the world, he had applied for a technician position. 2 weeks after he started, I asked him if he wanted to go on the expedition and take charge of the sampling. So right out of college, he came out, and this is the job he got. Just 5 years later, he just got off the boat briefly, so he's enjoyed it. What was expected is what we found for human diversity, like this 1 to 3% variation. But what we found was up to a 50% variation in these sequences. So on this graph, we can compare it to a complete genome sequence, and these little bars are roughly 500 to 600 letters of genetic code, and they're color-coded by different sites around the world where we sampled them. what we found is what people were measuring for diversity, 16S RNA, that basically these several thousand organisms with very diverse sequences all have basically the same 16S RNA. So scientists thought this was a single cell, not thousands of independent cells. So all this was missed. And this is what we find everywhere around the globe. It's absolutely been stunning with this. But as we add more and more data, we're starting to see at a global basis why people were misled. The data sort of collapses into major taxa that people thought were single species. So for example, SAR11 was thought to be just a single bacterial type. Now we know it's 5,000 to 10,000 different species. But they all come together at the sequence level with things in common. We've identified completely new ones with this. We can actually assemble the data, assemble genomes out of it. And this is just the very tip of the iceberg as we characterize the organisms around the globe. We've also gone very deep in the ocean and deep in the earth and had very different findings. So actually the third genome we did was, if I go back one, from this high temperature vent that's about 1.5 miles deep in the South Pacific Ocean. The temperature in the center of that plume is about 400 degrees centigrade. And the submersible Alvin broke off a piece of the chimney where the red arrow is, and this is the organism that was cultured out of that. It was named after the late expedition leader Holger Janisch. So this is an autotroph. We're not autotrophs. We actually have to eat organic substances. This makes everything it needs for life from CO2 as the carbon source. Its energy systems converting CO2 into methane. And it's driven by a hydrogen pump. So it's a pretty unique organism, and its temperature optimum is 85 degrees centigrade, which we would cook at. At our body temperatures, it's basically frozen. So it shows you the extent of diversity. We go much further now. We have organisms that go up to higher than boiling water temperatures, 115 degrees centigrade. We have organisms now, one from Northern California that grows in such strong basic conditions that if you put your finger in it, your skin would dissolve. We have others that grow in strong acid. So the diversity in the microbial world is truly stunning that's coming out of some of this information. When we go deep in the earth, we actually get a different answer. It's pretty stunning. We find the same extent of species diversity that we find in the ocean. In fact, there's probably as many organisms, as much biomass under the surface of the earth than there is in everything in the oceans and in the visible world. But here we only see that 1 to 3% genetic variation. So what's the difference? A mile deep in the earth, there's no UV radiation, there's no sunlight, and even limited radiation of other types. So actually a organism that we got the sequence of that came out of a coal bed methane well in Colorado was very, very closely related to an organism we got out of a volcano in Italy. So there's various arguments of whether the oceans have been completely wiped out by meteors in the past, and these deep surface reservoirs would be reservoirs of life on this planet. And without all the radiation, they didn't have the same rapid extent of evolution. So it's getting to be even more exciting as we characterize our planet a little bit further. We've actually doubled the number of genes known to science. We're about to do it the 4th time. When we started, there was only roughly 1 million genes from all biology known. Just our first study in the Sargasso Sea doubled that number. Now we're over 60 million genes. So a simple idea. It's important for young scientists to know just the idea of taking these samples off a sailboat and sequencing them. That's where most of the genes known to science now have come off from sampling off the deck of the sailboat. So where are we? Have we saturated our knowledge? Do we have a good sampling of Earth? And the answer is no. If we look at human genes, mammalian genes, we pretty much saturated discovery of those genes. So sequencing more humans won't discover more genes. But if we sequence viruses, microbes, small eukaryotes, even with this 60 million gene set, we're still totally in a linear phase of discovery. So any one of you could go out and take a sample from any of the surrounding waters here and discover tens of thousands of new genes in a single sample, new gene families, new proteins that people have never seen of any evidence of in science. So it's an exciting phase. So minimal life, talking about can we reduce it down to its minimal levels of the reductionist approach. We sequenced this genome as the second one we did in 1995, and it has the smallest genome of any self-replicating species, 482 protein-coding genes and 43 RNA genes. And so in 1996, some of you might remember, NASA scientists claimed they discovered fossil bacteria in a meteor from Mars. But a simple calculation showed that these little tiny bubbles that they saw were so tiny, you couldn't even get a single molecule of DNA or RNA or protein in them. And so eventually they ruled it as an artifact. But I started a lot of thinking about what minimal life forms could be, how small could it be to have a self-replicating species. So we started asking those questions. What is essential for life? Do we understand those components? It's a way of defining life, something science has been trying to do for a long time. Many years and many different experimental methods tried to answer this, but none of the existing techniques would work. So we decided the only approach would be to try and chemically make the entire genome, where we could substitute genes, leave ones out, and see if we could get life. As soon as we went that way, we had new questions. Would chemistry even permit us to make these large DNA molecules? And if we could make them, would it just be interesting chemistry? Or could we actually boot them up in a cell? And because this is such an important notion, I want to spend essentially the rest of the time walking you through the stages of what we've done here. And it was just almost exactly a year ago where we announced the first synthetic cells, where the chromosome was made entirely from 4 bottles of chemicals, from code right out of the computer driving the process. And that code booted up this new cell type. And so I'll walk you through that. We started this in the mid-90s, the idea of it. And it turns out the machines that synthesize DNA are not very robust. Most people that use synthetic DNA use it just for primers, for sequencing, or for PCR. The problem is, as you make pieces longer, the error rate increases. It's an n-1 situation. The longer you make it, the more errors. And so we knew we had to develop new techniques for correcting the errors in the sequence. And so we decided to try this with a small virus. Phi-X174 is a virus that kills the bacteria E. coli. And it was also, historically, it was the first DNA virus to have its genome sequenced in 1977 by Fred Sanger and his team. Clyde Hutchinson, who was on our team, was a postdoc in Sanger's lab at the time and took part in sequencing that, and actually had a sample that he'd been carrying around in his pocket all this time. So we could compare the original species with new techniques. But we started with this code in the computer, and we, through these various synthetic steps, put together this 5,000-letter piece of synthetic DNA, sequenced it and checked it for accuracy. And the next stage was pretty exciting. We took this piece of DNA and put it in bacteria E. coli. The E. coli cellular machinery recognized the DNA as normal DNA, started to read it, started to make the proteins it encoded for. Those proteins self-assembled into the virus, and the virus showed its gratitude by killing the cells that made it. that's how we detect them. The clear packs on this lawn and E. coli is where the virus actually killed the cells. So we call this a situation where the software is making its own hardware. DNA is the software of life. We just put this chemical software in the cell, and we got this viral particle made that actually had tremendous biological activity. Now, our goal wasn't to make a simple virus, it was to make a much more complex bacteria. But we thought we can now at least synthetically make accurate viral-sized genomes. So if we made a lot of those pieces, we could then find a way to try and stitch them together to make the whole chromosome. So that's what we set out for the next several years to work on. We designed these 101 pieces that were 5,000 to 7,000 letters long, and started out to make these from pieces that were initially only 80 letters long. So we assembled these like we did the virus, and then we had to find ways to put all these together. Just to show you, at the lab level, this is conceptually pretty simple, even though you have to do it conceptually. You can't see what you're doing. You can see the result of what you're doing. So we designed these pieces so they overlap, in this case by 200 letters of the genetic code. And we have these enzymes that just chew back on one of the strands of DNA. Now, because there's this nice overlap, these pieces will spontaneously go together. It's a process called annealing. And then we have other nice enzymes that just fill in and repair that little gap in the DNA. And all of a sudden we have a piece now that's twice as big. So we started that in this, looks like a Final Four basketball playoff, where we put pieces together and got up to 24,000 letters of genetic code. At each stage we would grow these up in E. coli and sequence them and verify their accuracy. And then go on to the next stage and do this again. And we put 3 of those together, had pieces that were 72,000 letters, and checked those again. So this was a long, laborious process. We had a pretty sizable team doing this. And then when we got over 100,000 letters, these 144,000 letter pieces, half of them wouldn't grow in E. coli. We exceeded the limits of what molecular biology E. coli could deal with. So we had to find a new system, and we've been studying a number of these as we went. One of the most fascinating organisms is called Deinococcus radiodurans. You can find it probably in this room. You can find it now on the surface of the space station. This organism can take 3 million rads of radiation and not be killed. Don't try that at home. I don't think you'll survive even a tiny fraction of that. But what happens is, you can see at the top, its chromosomes, its DNA get blown apart into these little tiny pieces. But over 12 to 24 hours, when this sits in a water solution, it reassembles its chromosomes exactly as they were before. And once it does that, it can start growing and replicating again. So we have organisms on this planet that can take huge doses of radiation, which is when people talked about panspermia, like the late Francis Crick did, that maybe that's how life evolved here. It was sort of dismissed because no organism could survive the radiation levels in space. Well, it turns out we have lots of organisms on this planet that can. It's not clear they necessarily evolved to withstand radiation. These same processes are when these species completely dry out in a drought, and then water comes back in. They can repair all the damage done to their DNA, and they can come back to life and start replicating again. So we worked on this system for years, trying to see if we could get the system working independently of the cell, and we never could. But fortunately, there's a system that everybody here is much more familiar with. It's called brewer's yeast. You make beer and bread with it, and it has these tremendous capabilities of repairing and assembling DNA. So we just had to take these synthetic pieces that were 144,000 letters each and put them into yeast. And we added a cloning vector that I'll tell you more about a little bit. It just has a artificial synthetic yeast centromere in it. We put these 5 pieces together, and yeast automatically assemble this into the entire circular chromosome. So that led to our announcement in 2008 that we now had synthesized a complete bacterial chromosome from these 4 bottles of chemicals. As I said, it was a long, multi-year process, and we continued to work on the synthesis. We hired a young scientist as a postdoc named Gibson, who came from Buffalo, and pretty rapidly he invented a whole new technique and said, I don't know what took you old guys so long to do this. And it's a nice single pot reaction where you just mix everything together at 50 degrees centigrade for 15 minutes, and it all assembles. Because I don't know why you wanted to work for years. Why didn't you invent that? So, in this process, it's a little bit technical, but it's that process I just showed you where there's the exonuclease enzyme that chews back on one strand, and then the pieces anneal together. But the beauty of this is its simplicity. The exonuclease would just keep chewing away on that strand if you didn't kill it. But that enzyme is very unstable at 50 degrees centigrade, so it only works for a short while, then it dies. And the pieces go together, and this tag ligase and the polymerase just ligate it together and fill in that gap. So we just throw in all the pieces with the right design overlaps, and it puts it all together, which means we can now design an automated system to do this. They improved this by synthesizing the entire mouse mitochondrial genome using this very simple method. There's contests every year for students to take part in doing synthetic biology, and some of you may actually take part in this. They love this technique, and someone made a really nice video that you can get download from YouTube about how they were saved by the Gibson method. It's a nice musical. If you're a scientist. So we can now automate this. We can just start with the digital world, go right into robotic synthesis without human intervention. And the importance of this will become clear as we try to scale up at the next stages. So how do you boot up a synthetic chromosome? DNA, when you get it in these large pieces, is very brittle, it's hard to handle. We had to develop all these new techniques for moving it around. You can't just pipette it like you can other molecules. We have to move it around in gel blocks, move it in and out electrically out of the gel blocks. So trying to get so we could do transplantation of this genome into a cell was a real challenge. And, so this study that we published in 2007 is actually, I think, philosophically one of the most important ones ever to come out of a research groups. Because this is so important, I'm going to walk you through the details of it. But basically, by changing the DNA software in the cell, we completely convert one species into another. So it's pretty stunning, and you see it in real life. So let me walk you through that. So we have 2 microbial species we used in Mycoides and Capricolum. They're roughly the same distance apart genetically that we are from mice, on the order of 10 or more %. So at the time, we didn't know whether proteins would be required for transplantation and booting up. So we treated the chromosome we isolated from the Mycoides cells with proteinases to destroy all the proteins. If we're making synthetic chemical DNA, we want to know that naked DNA would actually work. We had a couple of gene cassettes to this chromosome so we could select for it, and so that if it got activated in the cell, it would turn the cell bright blue, making it easy to detect. Then we inserted this naked DNA into the recipient cells. We have this extremely sophisticated movie to show you what happened. So we inserted the chromosome into the cell. So now we have a cell species with 2 different sets of DNA software. And it's pretty exciting what happened next. So my friend and colleague in all this work, Hamilton Smith, got the Nobel Prize in 1978 for discovering restriction enzymes. These are the enzymes that cut DNA at very precise points, and we wouldn't have modern molecular biology without those. What we think happened is this new chromosome we put in the cell started to be read by the cellular machinery, and proteins started to get produced. And some of the early proteins that were produced were restriction enzymes. They recognize the other chromosome in the cell as foreign DNA and shoot it up. And so now we have the body of one species and the genetic software of another. So what happened? In a very short period of time, we had these bright blue cells. And when we sequenced all the proteins, there was no evidence that any traces of the Capricorn species were left. All the proteins were coded by the new chromosome we put in the cell. So it's a very dynamic process, and this tells us a lot about how life works. So just simply by changing out that DNA, all the characteristics of the first species disappeared. And it was just what was encoded for by that new DNA that turned the cell into a new species. Life is a DNA software system. You change the software, you change the species. That's not how biology is currently taught. But it's changing pretty rapidly because this concept is changing the instructor's DNA as well. So it's very critical, and also shows the dynamic level of things. If you had a car where you had to replace 10% of the parts every hour or so, it'd be pretty frustrating. I think they're called Toyotas. But that's how life works. We constantly renew on an hour-to-hour basis, a minute-to-minute basis, all the proteins in our cells. So it's not like you make them once. You know, we look like we're kind of static. We were made once and that's it. We're constantly renewing them, making new components as the other, cellular proteins die off. So you put in new software, it immediately gets read and starts making these new proteins, replacing what was there. So some of you might recognize we have a problem at this stage, is that we actually assembled the bacterial chromosome, or prokaryotic chromosome, in a eukaryotic cell, in the yeast cell. So to do the same kind of transplantation that we just did with the native mycoides chromosome, we have to find a way to get it out of the yeast cell and transplant it into a bacterium. So to work out how to do this, we developed this new technique where we can actually clone entire bacterial chromosomes in eukaryotes. And they seem to be very stably maintained. In fact, it's pretty exciting, the discovery we made just by adding this small synthetic centromere. And when you look at pictures of your chromosomes, the ones that sort of look like a giant X or Y, it's that point that's at the center, that's the centromere. And so simply by adding a eukaryotic centromere to the bacterial cell, we can convert a bacterial chromosome into a eukaryotic chromosome. So now we have a system where we can try and isolate the wild type, the normal mycoides chromosome out of yeast and try and do the transplants. But we ran into a serious problem. It didn't work. And it took us 2.5 years to solve this problem and sorting it out. And it's somewhat ironic, given that restriction enzymes are involved in this. It turns out what happened was when we isolated the chromosome from the original mycoides cell, it had been methylated. So that's how bacterial cells protect their own DNA against the restriction enzymes. And so we proved this by isolating the genes for these methylases. We specifically methylated the chromosome that we were growing in yeast. And once we methylated that DNA specifically, we could then get the transplants to work. And we ultimately proved it by taking the genome from the recipient cell and we removed all its restriction enzymes. When it had no restriction enzymes, we could then transplant in native DNA. So the reason that initial transplant worked were the restriction enzymes made by the new chromosome. they recognized the chromosome in the cell as foreign DNA because it didn't have protection against those enzymes. When we just used naked DNA, the enzymes that were already in the cell immediately chewed up the synthetic or the DNA that we put in. So now we had a way where we could solve all this, and we published this paper in 2009 thinking we solved a lot of the riddles of life, where simply by adding the centromere to a bacterial chromosome, we could isolate it from the bacteria and grow it as a eukaryotic chromosome. Now, what's the significance of this? Many of you probably work on different bacterial species or have tried to. Most bacteria don't have genetic systems that allow them to be modified in the laboratory. That's why people study E. coli, for example, because it's easy to work with. But as soon as we put that bacterial chromosome in yeast, we have the complete repertoire of yeast genetics to make changes in that chromosome. Homologous recombinations, we just put in a similar sequence, designed properly, and it will immediately swap out into the chromosome. So we can just make these kind of changes very readily in yeast. So we can engineer it, methylate it if necessary, and then we can transplant it to a recipient cell to make a new engineered bacteria. So it's a very powerful tool that changes and gives us ability to alter organisms that were never possible before. But now we thought we'd solved everything in terms of being able to take the chromosome out of yeast and do the transplants. So we decided, we got kind of bold, we had these new methods of synthesis, and so we decided to make the much larger chromosome of the M. mycoides that we've been studying so much. So this is 1.1 million letters of genetic code versus around 500,000. The reason we chose the smaller genome initially, we actually thought chemistry was going to be the toughest problem to solve. And now that we had this new method, we decided to go for it and make this entire larger chromosome. And the way we did this was pretty logical. We made pieces that were 1,000 letters long. We put 10 of those together to make pieces that were 10,000 letters long. Put 10 of those together to make pieces that were 100,000 letters long. And then we put 11 of those together in yeast and assembled the whole chromosome. And so we went through all this. We knew how to transplant it out of the yeast cell. We were sure this was all going to work, and we went to do the transplants, and they wouldn't work. Now, some of you probably work on software. Software engineers have debugging software that tell them where there's problems in their software code. So we had to develop new biological debugging software to tell us where the problems were in our genetic software code. And what we did is we took the native chromosome and cut it up into 11 100 kb pieces, where we could substitute a synthetic one for the native piece and find out if they worked and where the problem was. We finally got up to where you could have 10 synthetic segments and 1 native segment, and that would transplant. So we knew there was a problem in that last segment. We sequenced it with new DNA sequencing technology, but it turns out there's a lot of systematic errors in some of this new technology, which is why we're still trying to develop it with the XPRIZE. So we went and sequenced it with the original Sanger technology, and we found 1 letter deletion in an essential gene. So 1 letter wrong out of 1.1 million letters, no life. So we corrected that error. We did the transplants again. Sounds like you need to plug in my computer back there. And we did the transplants on Friday, came in Monday morning, and there was a single colony, a single blue colony on the plate. It's now gone through over 1 billion replications. When we isolated the chromosome and sequenced it, it was the complete synthetic chromosome. Now, that's one of the challenges when you have a new area like this. You have to think of what could be an artifact that could mislead you. And we were worried even a single molecule of native DNA could fool us into cell out of billions of experiments, that it could have been a contaminant. So early on, we designed a way to watermark the DNA. And this is the gene map of the mycoides chromosome. So each one of these colored bars is a unique gene. And so we worked out places where we could insert additional DNA into this chromosome. Now, with the first genome we made, we weren't very creative. We just signed the names of all the people that took part in the synthesis using the single amino acid code. So there's a single alphabet letter code for the different amino acids coded for by the triplet DNA. And people were happy to point out we weren't very imaginative and we didn't put in things like one small step for bacteria. So we decided this time to get a little bit more creative, and we created a whole new code that we put in with 4 large synthetic watermarks. Now, in the first watermark, we invented this new code that enabled us to write the complete English language, not the Canadian version, and complete punctuation and numbers. Using genetic code where it created frequent stop codons so it wouldn't create new peptides that had biological activity. And we built in a web address into the genetic code. So we didn't tell people this, we just told them they had to solve it. And people, if they solved the code, they could read the rest of this, and they would find the web address, and they sent emails to it. But now that it's been solved by numerous scientists around the world, we can show this. So in addition to that new code within the code, we had 46 names now written, and we decided to add some quotations from literature. So we added this James Joyce quote, "To live, to err, to fall, to triumph, to recreate life out of life." So somehow that seemed appropriate. And then from American Prometheus, Oppenheimer's biography, "See things not as they are, but as they might be." And more important, Richard Feynman, "What I cannot build, I cannot understand." Obvious metaphor. When this became publicly known, we got a letter from James Joyce's attorney. You didn't ask for permission. James Joyce was dead. We didn't really know how to do that. And this comes under the fair use where you can quote an entire paragraph. So we kind of got past that, but then we started getting emails from a Caltech professor saying we had misquoted Richard Feynman. If you look on the internet, this is the quote that's there, and it's in a number of books. So he finally sent us a photo of Feynman's blackboard. And the original quote was, "What I cannot create, I do not understand." I think it's actually a better quote, so now we're going back and changing it to quote Feynman correctly. So I think that should hopefully satisfy the Caltech contingency. So what's next? What do we do with this? Now that the control experiment works and we know we can do this, I view these 60 million genes we've discovered, and that's going to be hundreds or 200 million genes relatively soon, as design components. Much in the same way the electronics industry in the 40s and 50s had resistors, capacitors, transistors, and integrated circuitry. We have 60 million of these components. It's actually too many to intellectually cope with. But it gives us a huge repertoire to basically make anything we want. We actually have computer software for designing new biological software. And that's what we're doing. We're trying to design new species, and basically you can build anything into these. You get them to do metabolically what you want. You can pick the type of metabolism, whether it's glucose metabolism or CO2 to methane. We can add safety factors like suicide genes, autotrophic markers. There's been tens of millions of molecular biology experiments with genes of every known species put into E. coli without any problems. Why is it? It's because the E. coli we always use in the lab has an autotrophic marker, a chemical dependency where it can't grow outside of that lab. So it's easy to build that in to the design of these organisms. So this is an exciting phase. But because of the sheer number of all these components, most of which we don't know what they do yet, we're developing these new combinatorial approaches to do very large numbers. So we're trying to take these robotics and get where we can make on the order of 1 million chromosomes a day. We don't have enough scientists on the planet to solve this, but just think, a single scientist building and testing 1 million different chromosomes a day changes tremendously, what human capacity is and can be. You just think that the numbers, if you only had 10 genes and you had 10 versions of each of those genes, that's 10 to the 10th combinations. So to vary 1 million or 1,000 genes in a simple genome, or 5,000 genes in something like E. coli, it's going to take just a tremendous effort. I actually think we can design and build a robot that can do all this in a combinatorial fashion and learn from the changes it's making, and learn thousands of times faster than any human scientist. So we actually have a chance of learning some of the biology on our planet by building a new technology. I'm sure this question's occurred to at least one person here. Why do this? And there's lots of different reasons, but I'll just deal with not the basic science ones, which hopefully those are obvious to you. Some of the implications for our society. We have roughly 6.8 billion people right now on our planet. I was born in 1946. There's now 3 people on the planet for everybody that existed the year I was born. So in my lifetime, we've tripled the population of our planet. Within most of our lifetimes, it'll be 4 times what it was the year I was born. So we're going to add in the next few decades much more than the equivalent of another entire China and India to the Earth. We're not doing a very good job of providing food, clean water, energy, medicine, without destroying our entire environment in the process. How are we going to do that when we have 9+ billion people? So I agree, science is not a nice thing to have. It's nice for medicine or something. We now live in a society that is 100% dependent on science for its future. So what are some of the notions? This is data from WHO. And when I first looked at these numbers, I was kind of stunned, because when you know somebody that dies, it's usually a big thing. There's 60 million people that die a year on this planet. So that's a lot of funerals. And you're going to see, the projections for the next few decades, it's going to stay pretty constant even while the population builds. In part, look at the projected decreases in communicable diseases. This is projected forward based on improved sanitary conditions, but largely on developing new antimicrobials and new vaccine approaches. The bad news for all of us as we all age is heart disease and cancer are expected to go from 63% to 76% of the population, so the deaths are going to go up substantially from that. Injuries, accidents remains pretty constant at around 10%. So there's a lot of implications in this. Obviously, some people have argued that to keep the population constant, we need to increase the death rate. I don't think that's a very viable, it's not a good approach. But you can also see even things like wars on these numbers of 60 million people a year dying don't really move the needle that much. So we have challenges of how to deal with these populations. So some of the new approaches that we're taking, for example, with vaccines, we've just completed with Novartis phase 3 clinical trials of a new meningitis B vaccine in Europe. That's pretty exciting. It's the first genomic-based vaccine. We designed this by sequencing, actually in the mid-90s, the meningitis genome. And and then looked for genes that were not subject to constant variation, like stable genes, and chose those to build as the vaccine candidates. So this work against all strains of meningitis B, and meningitis is one of those diseases. By the time you diagnose it in young men and women, you can't do anything about it. You can prevent the disease, you can't treat it once it's been diagnosed. These kids die too quickly. But it's a long time. It was 15 years to get to this point. We just announced forming a new vaccine company out of synthetic genomics and working with the U.S. government and Novartis, trying using our synthetic DNA processes to make flu vaccines very rapidly as a prototype test case. With H1N1, it took about 6 months to get the new H1N1 vaccine out, and a lot of people died in that interval. The first 3 months of that was just trying to get the seed stocks. Our goal is to be able to make the new seed stocks in less than 24 hours. So NIH has funded my institute to make synthetic pieces of every flu virus that's ever been sequenced, which is a large number. And so we'll have all these, like Legos, sitting on the shelf, that any time a new strain is discovered that we want to have a vaccine against, we simply assemble those together with some of the techniques that I showed you and create a new version of the flu virus that can go into some of these new cell culture methods that Novartis has. So our goal is hopefully within the next 2 or 3 years to be producing on large scale the flu vaccine that everybody will be using. And hopefully there won't be a new pandemic in that time, but these are techniques that allow us to deal with rapid changes very quickly. This is the famous Keeling curve on measuring CO2 in the atmosphere since the mid-50s. And obviously this curve was continuing to go up, in part because we've saturated all the CO2 sinks on our planet. The ocean, as you can see, is obviously the largest sink. But now that that's saturated, we're just accumulating more and more CO2 in the atmosphere. And obviously burning fossil fuels is the major component, but deforestation is not insignificant on its own. So the challenge is, how do you shift this equilibrium? Obviously shifting away from fossil fuels would be one way, but we're trying to not only shift away from fossil fuels, we're trying to use CO2 as our major source of carbon, trying to affect it from two sides. Agriculture is not very effective. I mean, obviously it feeds us today, but when you look at the actual yields from agriculture, take huge amounts of land, huge amounts of water, and the amount of food produced is not all that great. When you look at the oil produced by different plants, the best plant is oil palm. Almost 6,000 liters per acre per year. But with algae, even reasonable numbers can get us up over 10,000 gallons per acre a year pretty quickly. The U.S. has a very good corn lobby, and so we have a massive effort to make ethanol from corn sugar. You can see you get roughly 18 gallons per acre per year. So if we're trying to replace all the transportation fuels in the U.S. Based on corn, it would take a facility 3 times the size of North America. If we're trying to do it from algae, it would take a facility roughly the size of the state of Maryland, or what I've argued would be a wonderful use for Nevada. Still a big facility, but on the doable scale. So we've been working on designing new cells that get their carbon from carbon dioxide, their energy from sunlight. We're designing cells that take this into a variety of directions. I went to the major oil company meeting a few years back in Texas and announced that our goal was modest. It was to replace the petrochemical industry. And instead of shooting me or trying to feather me, some of them started to work with us. So we got $600 million in funding from ExxonMobil to see if we could actually convert CO2 using these cells into a new biocrude that could go into their refineries to make gasoline, diesel, and jet A fuel. So we have a pretty large team working on that. We just, this last year, opened a big greenhouse in La Jolla, right near the Torrey Pines golf course, where we're testing a wide variety of algae strains that we're picking up from around the world and ways to grow these. These are going to be facilities. Each facility has to be on the order of several square miles, so there's not going to be a trivial infrastructure. But actually, ExxonMobil is willing to invest the billions of dollars that we get up to certain levels of production to build these facilities. So, pretty elaborate testing with some modest success as we're going forward. So we're very optimistic about this approach. Now, some of you might have thought there might be some ethical concerns about trying to create new life forms, what they might do to the world. So we thought this was not unreasonable ourselves, and we asked, right after we thought of this approach, for a bioethical review. And the University of Pennsylvania Bioethics Department took this on as a major study, and they published their results in Science in 1999, saying they thought that we were taking reasonable scientific approaches. Their one major concern was the bioterrorism threat from these new technologies, and that is obviously still a concern. The Sloan Foundation picked up where they left off and funded my institute along with MIT and a Washington security think tank to look at the security issues. We spent a few years doing that with involvement from multiple government officials from around the world. And if you're interested in this report, you can download it from jcvi.org. And basically, a lot of the security people said that there's so many ways to get dangerous agents. Find a dead cow on a farm and get anthrax from it, or that people won't necessarily go to these complex scientific approaches to make them. I'm not sure how comforting that is as a message. But these have been, fairly limited as threats, at least to date. And when we made our announcement last year, President Obama immediately asked his new Bioethics Commission to take this on as their #1 mission. And they did, and held multiple hearings. And they published their report last December, where they said this is such a critical area that obviously needs to be watched closely. They didn't think it needed any new laws at the moment, but they really wanted to advance the research for the ways to limit the survival of synthetic cells outside labs or production facilities. Picked up on the notion that we had started of watermarking the DNA, so we can clearly tell a synthetic species from a naturally occurring one and not confuse evolutionary studies. And companies that make these synthetic pieces of DNA should start being asked to screen them to make sure nobody's trying to use them to make infectious disease agents. 2 years ago, the Royal Academy of Engineering in England took on a look at synthetic biology, concluded that it's likely to be the #1 wealth generator for nations for the rest of this century, which is obviously a big demand for things, but we're trying to drive things in that direction. We've now shown at least that it's theoretically possible. We can put all the pieces together, and we're trying to now do this with some exciting new approaches to food and to energy, basically writing new software for life and using the dynamics of what we know about living systems now to create things that wouldn't evolve on their own. You wouldn't expect a cell to evolve to produce huge amounts of a lipid hydrocarbon. So we have to use our knowledge of genetics to get things to happen at 1 million times times the scale they would naturally. But we do have the tools for that, and it's an exciting new era where I think we're primarily limited by our imaginations. Thank you very much.
Provocative and informative and quite stunning. This segment, the question period, will be moderated by Charlie Smith, who is senior editor of the Georgia Strait magazine. Charlie is an accomplished career journalist. His career began in 1986 as a social producer of CBC Radio 1 in Vancouver. In this period, he also worked for the Georgia Strait, and in 1991, he became a staff writer for the Strait, working on the city desk and writing a weekly column called Strait Talk. Fortunately, he still writes that column 20 years later. He is an accomplished moderator, and I'm very pleased to have him with us tonight, and he'll explain the procedure for the discussion. Thank you, Charlie.
Thank you. We don't have a lot of time with Craig Venter tonight, so I asked that you keep your questions concise and appreciate the broadcast. I'll start with a couple of questions, and then if anyone wants to go to the microphones or on both sides of the room, they can line up and be prepared to ask some questions. So, sitting here in the dark. Anyway, so Craig Venter asked if it's possible to lift the house lights. So I'll ask the first question to get things rolling. ExxonMobil, you mentioned, has invested hundreds of millions of dollars into your research, and I'm wondering, what are the possible consequences of these synthetic cells in connection with fuel? And one of the reasons I ask is the United States currently, we go through 20 million barrels of oil a day, and about 12 million barrels per quarter. What's the prospects that could mean the United States off imported oil?
Theoretically, it could be possible. We can't produce billions of gallons just by our crude. It's not going to have any impact. So that's asking an awful lot of these new biological systems. But the teams that have been working now are making some tremendous headways, and the engineering becomes as important as the biological engineering. So the facilities to do this, to deal with this massive amount of cells, liquid, recycling the water, getting rid of the waste, all these are components of it, but doing this on multiple square miles is a big challenge. So we're saying that the first time somebody would likely have fuels in their tank from this process is probably on the order of 10 years. It's going to take years just to build these facilities. So, but we're in the process of trying to scale up to see.
And what would it mean in terms of the accumulation of carbon dioxide in the atmosphere? We're now, your chart was 390 parts per million. People say we get to 450, we're going to start frying. Can you reverse that?
Well, so it's not clear whether you can reverse it, but certainly by stopping to take the carbon out of the ground and burning it and adding it to the atmosphere, we can shift it two ways. Presumably we'll be able to take less out of the ground, but it's going to be a challenge for governments. Because if we're successful, oil prices will drop. We saw this in the 80s when everybody started to make alternative energy. Oil prices were really cut. All those industries were wiped out.
And less Canadian tar sands oil goes to the United States.
Yeah. So the challenge will be the role of governments to put a price on carbon. Because obviously, as you said, the consensus is we can't keep adding this indefinitely to the atmosphere. So not only can we shift it by taking less out of the ground, we can capture back some CO2. But when we burn these new fuels, that'll create CO2, so we'll be recycling it. So it's not like we're taking a net amount out of the atmosphere. The goal is just to stop putting new CO2 in the atmosphere.
I was just wondering if you could tell us about the insights that your research has given into maybe the necessity of what was like considered junk DNA in cells. So like by finding the minimum necessary genes, what does that tell us about junk DNA?
So what he's referring to is when we found in the human genome, only about 3% of all that DNA actually codes for proteins. Our brilliant colleagues called the rest junk DNA. They thought it was just useless information. Sidney Brenner said there's a difference between junk and rubbish. Said you store the junk in your attic, the rubbish you throw out. You saw from the map I showed in the mycoides genome, it's almost wall-to-wall genes. So small organisms are much more efficient with their DNA. But some of the biggest discoveries in the last decade in biology were findings of what other people call the junk DNA. All these regulatory microRNAs, for example, all came out of those regions. The structure of the chromosomes, the mechanisms for how they unfold for reading the genetic code for making messenger RNA, this is all a part of it. So I've never been willing to have my junk DNA removed, but I've offered to do that for some of my colleagues.
Next question on my right.
So I've read your autobiography. I'm a big fan. You're sort of known for being a maverick and doing things your own way and having sort of disagreements with the establishment of bureaucracies and universities and things like that at the NIH. But are you aware of sort of this homegrown DIY bio movement in California and like these home PCR kits and gel electrophoresis kits? How do you feel about that? Does that mesh with the security concerns that you mentioned before? Is that something to be encouraged? I mean, the Feynman quote is, what I cannot build, I do not understand. Should people at home be encouraged to build these things on their own?
No, it's actually a very important question, and some of my colleagues really encourage DNA hacking, as it's called, and for people to do this in their kitchens. I don't recommend people try and make new versions of flu virus in their kitchen. I don't think that's a good thing. You know, it's one thing in the computer area to be the first one on your block to make a computer virus. You don't want to be the first one to kill and wipe out your neighborhood. So I think we need a different set of regulations. These are very powerful tools that give individuals the ability to do things that would have taken armies in the past. And so we need to find intelligent ways to use it and monitor it. That's why some of the regulations were proposed of the companies that synthesize the small pieces of DNA. Then somebody couldn't call up and order the smallpox genome pieces and make that in your garage. So, experimentation is a very important part of discovery. If it can be all that energy channeled in the right direction, that's with this iGEM competition. That's really done that in a nice fashion.
Great.
Is there like a book or a website where you've got all this information that you just showed us and compiled so you can see it at home?
That's a good one, and you can see most of it at home. You can start with my institute website, it's jcvi.org. And most of the papers that we publish are in journals like PLOS, so you can just download the papers from the internet. So our synthetic cell paper, Science made available, we can just download that off the internet. So search under synthetic life, synthetic DNA, and you'll find some pretty cool stuff.
Next question on my right.
Hi, in your yacht research, I'm wondering how close you are to finding a limit in genetic diversity.
We found out with the crew. In terms of the organisms out of the ocean, as I said earlier, we're in a linear phase of discovery. The viral world is barely understood now. One thing I didn't mention, it's actually a little diabolical finding in the viral world. They actually have super versions of bacterial genes that the host bacteria that they infect, they actually update and correct the genes in those organisms so they can stay robust and make a lot more virus. So it's not clear where the hierarchy is out there, but these are some of the findings that are just starting to come out. And, we're supposed to be looking for life on Mars and elsewhere in the universe. You can make more major discoveries by taking samples of low tide out here or just samples around the environment. You can make major discoveries of these things.
Thank you.
I wanted to see what your ideas about individuals having access to their whole genome and the idea of personalized medicine for the future.
So, as you might imagine, I'm a strong believer that you should have access to all your own genetic and medical information. I think people are just slowly catching on to that in the world. A lot of people were very afraid to have anybody else look at their genome because all their inner secrets would be revealed. I can tell you that far more revealing to write an autobiography. It tells much more about somebody. In fact, that's why at the time I chose to do my genome, is because there was so much fear out there that I knew I could give myself permission and understand this. And people are finding it's not so fearful. Jim Watson was the second one to have his genome done, and now people are rushing out to get their genome on the internet. And it also depends on your political system. We have a health system here. The United States, with President Obama leadership, just caught up to the modern world. And now, with health coverage being available for everybody, there's no preconceived notions about pre-existing conditions. So that was the worry. If you discovered something in your genome that you were likely to get a disease later in life, insurance companies wouldn't cover you. Now in the U.S. they have to. So it really depends on the society, what the rules are, whether there's any dangers. In terms of personalized medicine, there's not going to be medicines made for you or me specifically, but our genetic codes will tell us our risk for disease and our risk for having adverse reactions from different, for example, pharmaceutical treatments. Most drugs only work on about a third of the population. They do no damage to another third, and the final third, they can have negative consequences. You'd certainly like to know where you are with those. Your genome will tell you that in the future. So it will tell you how to prevent diseases. But those are different approaches that we take to medicine right now.
Question on the right.
Hi, Dr. Venter. As we approach the 1,000 genome, I'm curious to know if you can foresee what the next technological bottleneck might be and what the next XPRIZE and biotechnology tools might be.
So it's a very good question. So our ability to read the genetic code and the ability to do something with the data sort of go hand in hand. I had a nice argument with Gordon Moore about his Moore's Law. And the way to change it. DNA sequencing is changing faster than Moore's Law. Is it yes, but you're dependent on computers to analyze the data, so Moore's Law trumps nothing. And that's largely true. We're generating more and more data now faster with these machines. So we need bigger, faster computers to deal with it. So that's going to be one aspect. Data transmission is even a bigger problem. So my institute is on both coasts. We had a dedicated optical fiber for moving data back and forth. The new machines generate so much data, it's too slow to send it by optical fiber now. So we're back to sending it by FedEx. They move large amounts of it. The real challenge is, we had some nice discussion of these today on this, is characterizing human phenotype. The reason we can't tell very much about my genome or anybody else's sequences, we don't know how to interpret it. And so we writing everything down about your life, your chemical structure, your biology, your clinical lab tests from everything else, how your brain functions, how your memory works. In a digital form where we can compare that to your genetic code and tens of millions of little. That's where we have to be. So we have a lot of advances that need to take place before we get to preventing disease.
So we've heard a lot about the scaling up tonight and how these things happen way faster than anyone thought possible. Lots of doubling. This is kind of a perfect lead-in. So I guess from the practical end of how do we make it happen to the crystal ball end of what miracles do you see coming of this? And when we move from solving small problems like climate change to onto what these incredible feats of the next century, where do you see this going?
Well, I had a fun invitation a month and a half ago to take part in a NASA and DARPA study on the 100-year Starship project. Trying to actually design the Starship Enterprise for visiting distant galaxies. And the problem is radiation. I mean, we're not going to caucus-radiated neurons. We're killed pretty readily by radiation. And shielding is too much of a problem. So I suggested we just send digital information where when these get centuries later to where they're going, they just boot it up and create the life forms on the spot.
We have time for one more question, and I'm sorry to cut it off, so
Oh, maybe.
we're running out of time.
I'm willing to take the last, or as long as nobody else jumps in line.
So we'll go to John Wright.
Okay, yeah, thank you. We keep on hearing about how Monsanto's implementation of this sort of technology, and I assume they're similar in some way, will increase crop yield and so on. But studies show that it hasn't. And what are your thoughts on how you're going to implement it and how it's going to, sort of trails on the last person's question, how it's going to make more food and so on, right?
So one way is just to get rid of agriculture.
So hydroponics?
So we look at 600 gallons per acre for oil palm versus 10,000 for an algae farm that can make palm oil. You could save a lot of jungle in Malaysia and Indonesia that's been burnt down now to plant for oil palm plantation with a much smaller facility. That's one approach.
I'm curious about the idea of the synthetic life forms and in terms of relating that to natural ecosystems in terms of world mutation, because I think that's where some of the power of the metaphor software starts to break down. So how do you start to actually investigate the ecological interactions of these synthetic organisms and the role of mutation that would play once you get these organisms to interact with each other outside of the lab?
There's multiple components to your question. They're all very important. I'm totally against putting synthetic life forms in ecological systems unless they're totally self-contained, closed ecological systems. 40% or so of the oxygen we breathe comes from algae in the oceans. We don't want to shift all that to making lipid soup instead of oxygen. I don't think that's a good thing. There's other species that would argue with us that would rather eat the lipid soup. So I think they need to be under these tight control systems, and they should not be where they could end up in the environment. And so people creating GMO algae, if they don't have these strict controls, shouldn't be doing them in open systems. The mutation rate is something we can actually control. That was a key thing behind what we selected for the meningitis B vaccine. It was one of the discoveries with the first genome was we found tetrameric repeats, so repeats of 4 letters in front of all the genes for cell surface antigens and homophilus. That's why if we did a sampling of everybody's airways here, everybody would have a slightly different strain if we sequenced the homophilus from each one of us. Because what these repeats do, the polymerase literally trips on them and knocks out genes downstream on a random basis, constantly changing the structure of the cell surface antigen. The bacteria do this as a way of trying to avoid our immune system. So when we're looking for stable systems for vaccines, we chose ones that had none of these mechanisms for increasing their rate of evolution, so they're very stable. So actually for an industrial organism, we do not want it to constantly evolve at a rapid rate. If you're doing experiments in the lab where you want to have fast evolution, we know how to make things go even faster. So it is something that's under genetic control, but what I showed you with UV radiation and other things still will cause mutation. And a number of population scientists want to see how long it takes for Feynman's quote to be corrupted. Does it take a few trillion replications, or what does it take? So they're all important issues. Thanks for the question. This will be a really profound one.
Dr. Bender, as we move towards the $1,000 genome, and as we see the emergence of personal recreational genomics, a largely unregulated or entirely unregulated industry thus far, how do you feel that the potential scientific resource of thousands and millions of people sequencing their genomes and making them available as a bioinformatic resource should inform the regulation of this emerging industry?
Yeah, it's a good question. So you did well on the book. I think companies like 23andMe and Navigenics, where you can get a lot of genetic information for a few $100s or something, they're tremendous education tools. I mean, if you're interested at all in your own genetics, for $400 you can get a genetic survey. Their website is a wonderful educational tool. The harm actually comes not from that process. It comes from bad genetic data coming out of the research community or driven by a commercial product. So, for example, back when the BRCA1 gene was discovered, companies selling this test for $500 a test would tell women if they had the genetic changes in the BRCA1 gene that they were better than a 90% chance of getting breast cancer. And so some women used this information out of fear. They went and had double radical mastectomies, thinking they were protecting themselves from breast cancer. The information they were given was just fundamentally wrong in the first place. And it would probably have been better just to have it randomly out there themselves instead of being fed crap by a non-knowledgeable physician. So the challenge is how to get good information out there, not saying that we have to treat society in a paternalistic fashion and limit the information they should have. We need to teach people how to interpret the data, and I think everybody should have access to it and have the right to do that.
Other things that you could have done this evening. We're very glad you spent it with us, and please keep posted for the fall event. Thank you very much.
You're kicking over everything.
Yeah, well, it's part of the winter life,
It's phenomenal.